Okay.
Dear students, in previous lectures we discussed about the somatic structures and asexual reproduction in fungi. In asexual reproduction we discussed that fungi reproduce by means of asexual means like conidiophores, conidia, conidiospores, chlamydospores, then fragmentation of hyphae, then budding. These all methods we discussed earlier. Now today we will discuss about the sexual reproduction in fungi and other methods of the reproduction, that is parasexuality in fungi. So you all know that sexual reproduction involves the union of two compatible nuclei, and the nuclei may be carried in motile or non-motile gametes, in gametangia or somatic cells of the thallus. So there are three phases of reproduction, sexual reproduction, that is three typical phases occurs in sequence during the sexual reproduction, that is plasmogamy, karyogamy and meiosis. In plasmogamy, anastomosis of either two cells or gametes and fusion of their protoplast take place, and in this process the two haploid nuclei of opposite sexes, that is compatible nuclei, are brought together, but the nuclei will not fuse. While in karyogamy, the fusion of two haploid nuclei brought together as a result of plasmogamy occurs, which is known as karyogamy. This stage follows immediately after plasmogamy in many of the lower fungi and may be delayed in higher fungi. In higher fungi, plasmogamy results in binucleate cell containing one nucleus from each cell. Such a pair of nuclei is called dikaryon. So you can understand the process of sexual reproduction, that is three distinct phases, through this very picture. What happens? In this case, the two hyphae of opposite nature, that is dissimilar type, having compatibility, will come closer, and then the plasmogamy occurs. So in plasmogamy, what happens? The two cell, the content of this cell fuse, and the nucleus of the two compatible nuclei come together in the fused hyphae, and the condition, that is n plus n stage, n plus n dash stage you can say, will appear, and this stage is known as the dikaryon. So in many cases after the plasmogamy and karyogamy, the meiosis occurs in lower fungi, while in case of higher fungi, in case of basidiomycetes, the extended dikaryotic phase occurs, and the fused hyphae in the later part meiosis occurs, and it remains for long, so it is known as extended dikaryotic phase in case of basidiomycetes. Now thereafter, meiosis, in meiosis what happens? There is reductional division, you all know, and after the fusion of the nuclei there is meiosis. As a result fusion of nuclei, the zygote is formed, and the meiosis occurs, and in meiosis the chromosome number is reduced to half in first division, and then the other division, the cell 2 to cell become 4. So another division is known as equational division. So as a result, 4 haploid cells are produced, and these spores can be dispersed and germinate into new hyphae. What we have discussed, that is in three phases, what happens? First is the plasmogamy, then karyogamy, and then meiosis. So meiosis, it is now clear that meiosis is the nuclear, after nuclear fission, which eventually takes place in all the sexually reproducing fungi, is followed by meiosis, and karyogamy results in the formation of diploid cell, that is 2n, and diploid nucleus, that is 2n, and meiosis reduces the number of chromosomes to haploid and constitute the third phase of the sexual reproduction, which I have earlier told you and described you. And in this case, the nucleus undergoes a reductional division, earlier told. Now, the organs involved in the sexual reproduction. What are the organs? So, you must have the idea that fungi which produce morphologically distinguishable male and female sex organ in each thallus and are called hermaphroditic or monoecious or unisexual. So, in a single thallus of a monoecious fungi, a single thallus of monoecious fungi can reproduce sexually by itself if it is self-compatible. And in a fungus, when the male and female organs are produced on the two different thalli, then it is said to be dioecious or bisexual. Normally, a single thallus of a dioecious fungus cannot reproduce sexually by itself, as the thallus is either male or female. The sex organ of the fungi, you all know, sex organ of the fungi is called gametangia. The singular is gametangium, and the sex cells are called gametes, and the mother cells called sex organs are called gametangia. If the gametes and gametangia are produced are morphologically identical, then or similar, they are called isogametes. If the gametes and gametangia produced differ in size and structure, they are called heterogametes. So, and the gametangia is known as heterogametangia. In the later case, the male gametangium is known as, when heterogametes are produced, then the male gametangium is called antheridium, and the plural is antheridia, and the female gametangium is called oogonium, and the plural is oogonia. The male gamete is known as antherozoids or sperm also, while the female is known as egg or oosphere. So now, what the kind of you have the idea about the kind of antheridium. So antheridium basically is an elongated club-shaped multinucleate cell, much smaller in size than oogonium, and are produced terminally or intercalary. There are different types of antheridium. They are monoclinous, then diclinous, then paragynous, and amphigynous. Monoclinous antheridium are those which arise anywhere along the oogonial stalk. The example is Pythium species, where when the antheridium arises from another branch of the thallus, then it is called diclinous. So in case of Pythium, both monoclinous and diclinous situation exist, and paragynous and amphigynous. What is paragynous? Basically, the tip of the antheridium is attached parallel to the side of oogonium, during the gametangial contact. Example is Pythium and albugo. The two genera is having paragynous antheridium, and amphigynous, when oogonium perforates the wall of antheridium and penetrates into it and finally emerges out at the other side as a special structure. While passing through the antheridium, while the fertilization is effected, on maturity the sexual apparatus consists of a balloon-shaped oogonium with a thick walled oospore at the top of funnel-shaped base formed by the antheridium. Example is Phytophthora. So amphigynous condition occurs in case of Phytophthora species. There are several methods of sexual reproduction, and that is 5. The following methods which the fungi employ to bring the compatible nuclei together for fusion is known as the methods of sexual reproduction. One is planogametic copulation, another one is gametangial contact, then is gametangial copulation, spermatization and somatogamy. In planogametic copulation, a planogamete is a motile gamete or sex cells, the fusion of two gametes, one or both of which are motile. If then it is called planogamete, and then the and if the fusion of the two gametes which are motile, then it is called planogametic copulation. This type of sexual reproduction is common in aquatic fungi. There are three different types of planogametic copulation, that is isogamous motile gametes, copulation of isogamous motile gametes, then copulation of anisogamous motile gametes, then heterogamous planogametic copulation. You can understand it by this slide. So you can see that as you know, the isogamous is the morphologically alike gametes. When fused together, then it is isogamy. When the morphologically distinguished or different gametes are there, one is small, other is big, then it is called anisogamy, and the third is oogamy. Okay, so you can have about the copulation of anisogamous. It involves the union of the larger gamete with the smaller one, and the resultant zygote is motile, and the zygote resulting from the isogamous or anisogamous planogametic copulation forms the resting sporangium. On further development, it functions as sporangium by differentiating the zoospore internally. And in case of heterogamous planogametic copulation, a non-motile female gamete, that is oosphere, is fertilized by a motile male gamete, and this results in the formation of oospores, resistant structure and resting spores. Oospores germinate and produce mycelium directly. So this is all the three planogametic copulation, that is isogamy, anisogamy and oogamy. Now the second method is gametangial contact, also known as gametangy. In this method, the male gamete, that is antheridium, and the female gamete, that is oogonium, come in contact, and one or more nuclei from the male gamete enter the female gamete, and the oogonium dissolve in intervening wall through a pore or through a fertilization tube. So when the antheridium comes in contact with the oogonium, at the place of contact the wall dissolves, or a small tube, fertilization tube is developed, through which the nuclear material of the male gamete, that is antheridia, passes into the female gamete, that is oogonium. So this occurs, and in no case gametangia actually fuse or lose their identity during the sexual act. Okay, so fungi in the order Peronosporales are the examples, and gametangial contact is also common in some Ascomycotina, where antheridia and female organs, that is archegonia or escogonia, may or may not be well defined. So you can see in gametangial contact what happens. The antheridia and oogonia come closer, and here also the antheridial wall dissolves, and the nuclear material passes into the oogonia. Now the third method is the gametangial copulation. So this is the process of fusion of entire content of the two mating gametangia. So this can be had through two methods, that is mixing of entire protoplasm of male and female gametangia, and second by isogamous copulation. So in first method, the two gametangia meet and the entire content fuse in the female gametangia, leading to the formation of zygote. The zygote forms the resting sporangium. The examples is Chytridiomycetes, that is aquatic fungi. Now in second method, that is isogamous copulation, the two morphologically similar gametangial hyphic come in contact, the wall at the point of contact dissolves and the content mix in the cells, thus form this result in the formation of zygospore. That is the case of mucor, rhizopus and other phycomyces. So in example you can see in case of first, that is gametangial copulation, the first method, the two strain, two hyphae come in contact and their wall material at the point of contact get dissolved and there is mixing of the protoplasm. And in second method, the two gametangia come in contact and the pore is developed and the entire content of the male gametangium passes into the female gametangium. Second is, now fourth method is spermatization. In some fungi like rust and bears numerous minute non-motile uninucleate male cells called spermatia. Spermatia are produced in spiral receptacles called spermogonia or pycnia. Insect, wind or water to the female gametangium carries them, which is usually a receptive hyphae or trichogyne, to which they become attached. A pore develops at the point of contact and this content of spermatium passes into the particular receptive hyphae and this results in plasmogamy and initiation of dikaryotic stage of the cell. You can see the spermatia reaches in first cases the spermatia reaches the receptive hyphae and at the point of contact the valve get fused and the nucleus passes into the receptive hyphae and thereafter the dicaryotization occurs in the cell. While in case of somatogamy, which we will be discussing, it is the mating of hyphae of opposite types. And here also the nucleus passes into the negative strain and as a result dicaryotization occurs. So in somatogamy, two sex organs are in somatogamy, sex organs are produced and no sex organs are produced and here the word no sex organs are produced and somatic cells function as gamete, that is somatogamy, and hyphae anastomose and nuclei of opposite mating type are brought together in one cell. The somatogamy is common in Ascomycotina and Basidiomycotina group of fungi. Now you can have the idea about some other cycle of sexual cycles, that is also heterokaryosis. So what is heterokaryosis? The phenomenon of existence of different kinds of nuclei in same individual is known as heterokaryosis. The individual which exhibit heterokaryosis is called heterokaryon or heterokaryotic. Heterokaryosis may arise in fungal cell or fungal thallus in four ways. First, by germination of heterokaryotic spores, which will give rise to heterokaryotic soma. Second, by the introduction of genetically different nuclei into the homokaryon, a soma in which the all nuclei are similar, that is homokaryon, and in which if the other nuclei of different genetic base is introduced, then it becomes the heterokaryon. Third step is by mutation, where a multinucleate homokaryon, the mutant nuclei subsequently survive and multiply and spread among the wild type nuclei. And fourth, by fusion of some nuclei in haploid homokaryon to form diploid nuclei and subsequently survive, multiply and spread among the haploid nuclei. Thus, in some fungi, it is possible to have different kinds of haploid nuclei in the same soma and a mixture of haploid and diploid nuclei. So this is basically the heterokaryosis. Parasexuality. You all know that the parasexuality was discovered in the University of Glasgow on Aspergillus nidulans by Pontecarvo and Roper in the year 1952. What happens? Some fungi do not go through sexual cycle but derive many benefits of sexuality through parasexuality. What happens in this? This is a process in which plasmogamy, karyogamy and meiosis, that is haplodization, takes place, but not at a specified point of time in the thallus or in the life cycle of an organism. So parasexual cycle is very important in Deuterobasities, where sexual reproduction does not take place, and some fungi which reproduce sexually also exhibit parasexuality. And since it was discovered in the year 1952 by Pontecarvo and Roper from the Glasgow University, it has been reported in number of fungi in Escomycotina like Cochleobolus, Basidiomycotina like Puccinia, Belamsora and Eustlago. The sequence of events in a complete parasexual cycle is as follows. First, there will be the formation of heterokaryotic mycelium, then fusion between the two nuclei will occur, then fusion between like nuclei may occur, or fusion between the unlike nuclei may also occur. The third step is the multiplication of the diploid nuclei side by side with the haploid nuclei. Both the multiplication of haploid and diploid nuclei may occur. Then the occasional mitotic crossing over will take place during the multiplication of the diploid nuclei, and we sort out. Next step will be the sorting out of the diploid nuclei, and then occasional haplodization of the diploid nuclei if occurs. Then again sorting out will take. We can sort out the new haploid strains. So by this we can get the parasexual organism. Methods of reproduction. Now some of the terms, that is anastomosis, then clamp connection, then sexual spores, what they are, actually we should know, and we will discuss it one by one. So what is basically anastomosis? It is basically an important cause for heterokaryosis, and anastomosis involves the fusion of hyphae of some species, movement of one or more nuclei into one or the other fused cells, and the establishment of compatible heterokaryotic state. So anastomosis brings about the establishment of compatible heterokaryotic state. A similar nuclear displacement may occur in the adjacent cell of the same hyphae by formation of clamp connection, through which the nucleus or nuclei from one cell move from the another cell. So clamp connection is basically very common in the Basidiomycetes. So what is clamp connection? Clamp connection is basically a mechanism found in the Basidiomycetes. It is a bridge-like hyphal connection, characteristics of the secondary mycelium in many Basidiomycetes, and it ensures that the sister nuclei arising from the conjugate division of the dikaryon become separated into two daughter cells, and clamp connections are found during nuclear division, and it is supposed to help in dikaryotization of the adjacent cell. You can better understand it through pictorial diagram. So in pictorial diagram, what happens? You say there is two different nuclei in the same cell, and there is a formation of clamp-like a structure where one nuclei move and other nuclei remain as such. Then the two nuclei divide side by side, having A that is having A dash also, and B is having B dash also, and there is then coupling, you can say, of the A with B and B with B dash, A dash with B dash, and as a result the dikaryotic mycelium dikaryotization happens, and in the next phase dikaryotic mycelium appears. So now the sexual spores. Sexual spores are formed as a result of fusion between two opposite sex gametes. They are resting spores incapable of germination immediately after formation. The sexual spores are oospores, zygospores, ascospores, basidiospore, deriving their names from the class to which the fungi belong. So if it is oomycetes, then it is oospores. If it is zygomycetes, then the sexual spores will be zygospores. If it is ascomycetes, then the sexual spores will be ascospores. And if the family, if the division is Basidiomycota, then the spores, sexual spore belonging to Basidiomycota is known as basidiospores. So now, what is oospore? Oospore is a sexually produced spore which develop from unequal gametangial copulation or markedly unequal gametic fusion. It is the characteristics of sexually produced spore of oomycetes. Earlier I have discussed, and oospores develop from the fertilized oospheres. Now zygospores are sexually produced resting spores or structures formed as a result of plasmogamy between the two gametangia, and which are usually equal in size. They are resting structures. Zygospores are typical sexually produced spores in zygomycetes, that is mucorales and entomophthorales. In mucorales, that is mucor and rhizopus. You can you will have the example. So in figure also you can see the zygospores and oospores. Now ascospores and basidiospores. What are ascospores? Ascospores are characteristic spore of large group of fungi known as Ascomyctina, which I have earlier told you, and they are formed as a result of nuclear fusion immediately after meiosis, and the 4 haploid daughter nuclei can divide mitotically to give 8 haploid nuclei around which the ascospores are cut out. Ascospores shape varies from globose, oval, elliptical, lemon-shaped, sausage-shaped, cylindrical or needle-shaped. The ascus is a sac-like cell, generally containing a definite number of ascospores, usually 8, formed by free cell formation, usually after karyogamy and meiosis. While basidiospore is a spore born on the outside of the basidium following karyogamy and meiosis. So you all know that ascospores are endogenously produced, while the basidiospores are exogenously produced. So in shape also they vary from globose, sausage-shaped, fusoid, almond-shaped, that is flattened, and the wall may be smooth, ornamented with spores, ridges or folds. So you can see here that the ascus, in ascus usually 8 ascospores are there, and in basidium it is a bulbous swelling on which the 4 basidiospores are produced. So with this we end the chapter, and thank you for your hearing and be with us. Thank you.